my dear students, and welcome to Zenith Academy Online. My name is Shagufta Jaipuri, and today we are learning a very important topic of ICSE Class 10, that is genetics, some basic fundamentals. This is a part 1 of the video series for this topic. In this video lesson, we will be covering introduction of what heredity is, what genetics is, what are the variations, the law of inheritance, sex determination, genes and the alleles. Okay, in detail I'll teach you what alleles are, and also we'll be doing some applications of genetics. So let's go ahead. Now children, here you can see a family. Here you can see a father and a mother and their 3 children. So you can see that these 3 children look somewhat like their parents. Okay. So what is genetics? Genetics is the study of transmission of body characters from parents to the offspring. There is some transmission of body characters. You can see here the mother's shape is round face. So the daughter has also got a round face. You can see the face structure of son is quite similar with the dad. You can also see the hair color of mother is transmitted to these 2 kids, whereas the hair color of father is transmitted to this kid. Genetics deals with the study of this transmission, the study of transmission of body features from parents to offspring and the laws which governs these transmissions. So now let's go ahead and study it in detail. So heredity is defined as the transmission of genetically based characters from parents to offsprings, and these characters which have been genetically transferred are known as inherited trait. So the trait or character that is genetically inherited or passed down from generation to generation is inherited trait. Now children, the inheritable feature is called character. Now, what is the inheritable feature? The height of the parent, the hair color of the parent, the skin color of the parent. So these are the characteristics which can be inherited. Now the alternative forms of the characteristic. For example, the characteristic is hair color. So what are the alternative forms? Red color or brown color. So these are called traits. So the inheritable feature, that is hair color, is called a character, and the alternative forms of the character, that is whether the offspring will get red color or brown color, are called traits. Now children, this is a very important saying that like begets like. Now what does that mean, like begets like? It means that young ones look like their parents. Cats produce cats and not dogs. So the offsprings look like their parents. But also you should remember that all the siblings, you can see they look like their parents, but all the 3 siblings do not look alike. The offsprings are never identical to their parents. Some difference is always found in between them, however minute it may be. So these small differences among these individuals are called as variations. So these small differences which make every individual different is called variations. Now we will talk about variation in populations. So here you can see, you can identify them as women, you can identify them as Homo sapiens, that is human beings. So human beings as a species share many main characters or traits among themselves which identifies the species Homo sapiens. So we can identify that this is the species of Homo sapiens and this is the species of dogs. Okay. Yet various races or tribes look different in several features. Here you can see that all of them are humans, okay, but all of them have different features. Here you can see an Indian boy who looks very different from an African boy. A British boy looks very different from a Japanese boy. So you can identify them. So various races or tribes also look different and they have different features. Even within the same race or tribe, you can see the individual members of the population show differences. Here you can see that all of them belong to a same tribe, okay, a same race, but still all of them are different from each other. Now further, the smallest unit of population is family, and there also you can see the differences in their body features. So these small differences among individuals is known as variation. I hope you have understood what heredity is and what variations are. Now, who are the carriers of this heredity? chromosomes. Chromosome is made up of two chromatids joined with the help of a centromere. Okay, so these chromosomes are the one who carries heredity. We'll see that in detail. Now, in humans, there are 46 chromosomes which are paired. So, it becomes 23 pairs and each pair containing 2 chromosomes. So, what are homologous pair? Homologous is a pair of 2 corresponding chromosomes. Both the chromosomes should be of same size and shape. One is from mother and one is from father. Now, here this is a karyotype. Now, what is a karyotype? Karyotype is a chart in which chromosomes are arranged according to the shape and size. Right. So, this is a karyotype of a boy child. Okay. So, here you can see there are total 1 to 23 pairs of chromosomes. Okay. Now, in each pair you can see that both of them are similar in shape and size. Okay. One is from father and another one is from mother. Right, but here you will notice that the last pair of chromosome is not same in size and shape. Now we know that there are total 23 pairs and out of which 22 pairs are autosomes. Now what are autosomes? Each pair is for different characteristic, for skin color, maybe for hair color, for height and all other different characteristics that we see. But one pair, that is the 23rd pair, is a sex chromosome. So all these 22 pairs determines how we are. What does it mean? How do we appear? What is our behavior? Okay. So all these 22 till here, all these 22 pairs are autosomes. All the pairs correspond to different characteristic, our appearance, our behavior, everything. So it represents how we are. Okay. Now 23rd pair, that is the sex chromosome, this one will determine who we are. Right. So in the case of a boy, you can see one is big, one chromosome is big. Here you can see this chromosome is big and this one is small. So the last pair will determine who we are. So if I see a karyotype like this, it means that this is a boy. Right? And if we see here the 23rd pair, here you can see that both of them are same. Okay. So both of them are X and X. So this is for a female child. So this last chromosome determines who we are. When we see this pair, so this determines this is XX, so this is a female. Whereas here it was XY, which means that is a male. So the 23rd pair, that is sex chromosome, decides who we are, that is whether we are male or female. Children, we have seen many a times that females are being blamed for giving birth to a female child or a male child. But that is not so. Okay. The decision of whether a male child will be born or a female child will be born depends on the chromosomes of a man, that is of a father. So here you can see that father carries, see, if we draw the germ cells of a father, that is the sperm of a father, so here it carries a chromosome X and this will carry a chromosome Y, right? Whereas the eggs of a mother will always carry chromosomes X. That we already saw in the karyotype diagram. Here if you see this X, okay, of a father, if it combines with, okay, if this combines with X of a mother, so XX, so now a daughter will be formed. Now if this Y chromosome of a father, okay, combines with the X chromosome of mother, then XY, that is a son is born. So who has determined whether son will born? The chromosome of father, because mother is anyways having only X. So if X combines with the X of mother, it is daughter. If Y combines with X of mother, it is son. Now again, if this X combines with the next X, XX, that is daughter is formed. And if that this Y combines with the this X of mother, it will form XY, and again son will be born. So you have seen that since female carries only XX chromosomes, whereas a male carry X and Y chromosome, so it is the father who should be responsible for the sex of the child and not the mother. Hope you understood how sex of a child is determined. So it's totally a game of chance. Now if this is an ovum of a mother, okay, which is X always, and this is the sperm of a male. So it is totally a game of chance that whether this will combine with the ovum to give female child or this will combine with ovum to give a male child. So it depends on which sperm fertilizes the ovum of a female to give rise to a baby. So now chromosomes are the carriers of genes. Now children, we have already studied that all species have a fixed number of chromosomes. Like human beings have how many chromosomes? 46, which are formed in the pair of 23. Similarly, all the other species have a fixed number of chromosomes. So they have a fixed number of chromosomes. However, the characteristics of species, including physical appearance, body function, etc., are not simply the outcome of chromosome number alone. Okay, so only the chromosome number will not decide that which character you will have, right? Because all human beings have same number of chromosomes. So what will decide what are your body characters? So the body characters, physical appearance, our behavior, all are the result of units called genes, which the chromosomes carry. There is something known as genes, a small unit known as genes, which is present where? In the chromosome. And these genes are located on the chromosome. So if you see here, in the cell, there is a nucleus, and in the nucleus, can you see this chromosome? And the smallest unit of this is a gene which is there in the DNA. So that gene determines what characters we will have. We'll talk about this in detail. Now let us see genes and the alleles. So here pair of chromosomes are not same. All the pairs of the chromosome. So when we saw the homologous pair, okay, which is same in shape and size, but these pair of chromosomes are not exactly same. They are similar. I told you they are similar in shape and size, but they are not identical. Okay, they contain version of the same genes, which is known as alleles. Okay, so what are alleles? Alleles are found at the same spots on the chromosome. So if you see this same spot, so this is an allele for which character? Hair color. So if you see here, these alleles are found at the same spots on the chromosome at the same location, which is called as loci. So it is found in the same location, but these two are different. So this is from, say, mother had red color hair, and this is from father. Okay, so red color was from mother and brown color of the hair was from father. So there are presence of two alleles, okay, in the same chromosome at the same position. So children, in a pair of homologous chromosome, one is inherited from the male parent. So one is inherited from the male parent. So this is known as a paternal homologue, and this is a maternal homologue, that is carried from the female parent. Here you can see at a position these two alleles are present. So what is this position or a fixed point called? A genetic locus is the location of a particular gene on the chromosome. So this is the location of a particular gene, and this is known as a genetic locus. Now at each genetic locus, an individual has two alleles. We saw in the previous diagram that at each position there are two alleles. Okay, has two alleles, one on each homologous chromosome. Now you can see here I have shown three gene pairs at three different genetic locus. Okay, here you can see two capital A's. So capital here in genetics denotes dominant. Okay, so I will tell you what dominant and recessive is later on. Here you can see these are the same type of alleles. So that is why it is known as homozygous. Okay. Now here you can see again both are same type of alleles. So homozygous. Homo means same and zygous means type. Okay. Now this here you can see here there is a capital C and here there is a small c. So this is heterozygous. Now you can see both are capital, so it is dominant. Both are small, so this is recessive. And here one is capital and one is small. Okay. I'll explain you about dominant and recessive later on. First you should understand that at one position, at the same position, that is at a genetic locus. Okay. At a particular position of the chromosome, there are two alleles, okay, of the same gene. Okay. So for example, it is of colors and two alleles of the color is present, red and brown. Now children, since we understood that there are two alleles of the same gene, so if on the chromosome of a child there are two alleles, okay, one is for brown color and one is for red color, then what will decide that the offspring will have brown color or red color? See here you can see that mother was having red color, father was having brown color, and in the chromosome of a child there will be both present, this one from the mother and this one from the father, red and brown. Now what will decide whether the baby will have red or brown? So there is something known as dominant and recessive alleles. So here we have taken an example of blue eyes and brown eyes, and in this character, okay, of eye color, always in all the character, one character is dominant and one is recessive. Dominant means in the presence of that character. For example, here it is brown eyes. So even if the gene, the allele of blue eye is present, then also the dominant one, the gene that will be expressed will be the dominant one. Okay, I'll explain you again. If there are two genes, two alleles of a genes, one is blue color and one is brown color, so always out of these two, one is dominant, that is in the presence of the other also this will show the characters, this allele will manifest. Okay, so that is known as a dominant one, okay, which will show its presence even in the presence of this recessive. So here I have taken an example of three individuals. So you can see in individual A, here if you see this allele, okay, it is present at the same locus, at same position, two alleles of the same gene are present. Here you can see that blue recessive is there. So if I take small b to represent this, and this is capital B. So what is the genotype, that is genes that we are seeing, that is capital B, small b. Okay, so since they are different, it is known as heterozygous. Okay, now here if you see here, you can see that both are brown, and since brown is dominant, I take capital B for both. Now what is the genotype of this? What are the genes? That is B and B. Okay, so this is homozygous because both are same. Okay, now here if we see both are blue, small b, small b. So since both are blue here, what will be the color of eye? Blue only. Okay, so this is what is the genotype? Small b, small b. So this is also homozygous, but here what is dominant? This. So the individual A will have brown color. Now here both are dominant, so obviously it will have brown color. Here both are recessive, there is no presence of dominant, so this will have blue color. Okay, so this is homozygous, recessive, both are same, homozygous and both are recessive. This is homozygous, dominant, and this is heterozygous with the color brown. So what is the genotype of this? Capital B, small b. Here the genotype is capital B, and here the genotype is small b, small b. But what is the phenotype? Phenotype is the physical expression that you see. Here it is brown, here it is brown, and here it is blue. So the ratio is 2:1. For brown is to blue, if you see for brown is to blue, what is the ratio? 2:1. So this is known as phenotype ratio. I will explain you phenotype again, but since we have taken an example here, I will show you that how to take out the ratio. So the phenotype ratio is 2:1, and what is the genotype ratio? Now you can see all three are different. So 1:1:1, all the genes are different. Okay. So let me explain to you genotype and phenotype again. The genetic constitution pertaining to the kind of gene possessed, which kind of alleles are present, are called genotype. Okay. So what are genotype? Genotype is the kinds of genes possessed. Okay. And what is expressed, that is what is shown, what we see. Okay. The seen character, that is the expressed character, is phenotype, because phen means to show. Now for the phenotype, what was the phenotype? Brown colored eyes. Last time we saw brown colored eyes was the phenotype. So for brown colored eyes there are two genotypes, homozygous dominant and heterozygous dominant. So here we can see which were the brown eyes, this one and this one. Okay. So for brown eyes, so brown color is the phenotype because we are seeing. So for brown eyes, what were the genotypes? The homozygous dominant and heterozygous dominant. Okay. So what was homozygous dominant? Capital B, capital B. What was heterozygous dominant? Capital B, small b. So these two are the genotypes, that is the genetic constitution. And what is the phenotype of these two genotypes? Brown color. I hope you have understood genotype and phenotype very well. So again I will tell you that genetic constitution, that is capital B small b or small b, whatever, the genetic constitution is known as genotype, whereas the physical appearance, that the gene that is expressed, that is what we see, is known as a phenotype, that is the color is phenotype. So for this, the phenotype brown, there are two genotypes present, that is heterozygous and homozygous. Now here students, I am taking an example of inheritance. So here I have taken a mom and a dad. So here I have taken an example that whether the child will be tongue roller or non-roller. What is tongue roller? A person who can roll his tongue like this. So a person who can roll the tongue like this is a roller, and a person who cannot roll his tongue like this is a non-roller. Here you can see a mom who is a roller. And which type of a roller she is? Here you can see capital T, capital T. Okay. So homozygous dominant. Okay. Now you should remember that in this case of roller and non-roller, always roller is dominant. Okay. So capital T, capital T. So mother is homozygous dominant, whereas father is a non-roller. So recessive thing should come. So small t, small t. Okay. Now this mom will have two sex cells, capital T, and father will have two small t. Right. So you can see that when this capital T chromosome, okay, and this small t chromosome combine, they will form capital T, small t. Now here the dominant is capital T, so the child will be roller. Okay. Now again this capital T will combine with this small t. Okay. So in this case also it will be a roller, because here one capital and one small t. Okay. So roller. Now if this t, okay, this t combines with this t, small t. Okay. You can see again capital T, small t. So again a roller. And if this combines with this now, it will again form capital T, small t. So in this case also roller. So all the four possibilities is of roller, and all of them, okay, all of them are showing which genotype? The same genotype. So the ratio of genotype is 1:1:1:1. Also the phenotype is of the same ratio, because all of them are rollers. All of them are expressing the same gene. Now in the same example we are taking another case. Now here you can see mom is roller, but here now you can see this is heterozygous, capital T, small t. So heterozygous dominant, and the father is again a non-roller. So she will have capital T, small t, and the father will have both small t's. Right. So now when these chromosomes combine, you can see one is capital T small t, another one is also capital small t, but two of them are non-roller, right? So you can see what are the genotype here. So genotype are what? Genes, okay? So what are two types of gene expression? Here you can see capital T small t is there and small t is there, okay? So both of them are 2 in number, so the ratio is 1:1. And what is the phenotype ratio? What is phenotype? Phenotype is the expression, okay, what you can physically see. So phenotype, that is roller and non-roller. So one is roller and another is non-roller. So now in this case, how many are roller? 2 are roller and 2 are non-roller. So 2:2. So here also the ratio is 1:1. So children, now let us study what is a Punnett square, because you will be told to draw Punnett square in your exam. So it is a simple diagram in which different types of gametes with the consent trait of one female parent are placed along one side of a square. So here if you can draw a square like this, here the one parent, that is maybe female, is here, okay, and are placed along one side of the square, and those of the other parent, that is male, will be placed here, are placed on one side of the square, and those of the other parent are placed along the other side. Then the possible combinations, that is genotypes of the opposite gametes, are given in the subsquares. So here suppose the female is giving capital T and male is giving small t. So now here the genotypes, that is this combination and this combination, like this is written. So then the possible combinations, that is genotypes of the opposite gametes, are given in the subsquares. The resulting phenotypes can be written under the genotype. So here what will be the phenotype? Here it is a roller. So this can be written like this. So we'll see an example here. Now here you can see a Punnett square where one parent is written here and another parent is written here. So this parent male is a homozygous tongue roller. Homozygous means same, RR. So one is having R, another chromosome is also having R, capital R. Now this is heterozygous tongue roller. So one is capital R, another is small r, because it is heterozygous, right? So now we'll form the combination. This capital R and this capital R give you capital RR. Okay, this is the genotype. And what will be the phenotype? What you can see here, this will be a tongue roller. Now here if you combine these two, again capital R, and the phenotype is tongue roller again. Now here capital R and small r, but since this is dominant, so again the phenotype is tongue roller. Here again capital R and small r, but since a dominant gene is there, so this also is a tongue roller. Now genotype ratio, if you want to write, genotype ratio is what here? Genes. So capital R is 2 times and capital R small r is again 2 times. So the genotype ratio is 1:1. Okay. Now what is the phenotype ratio? That is the gene that is expressed, that we can see that all of them are tongue rollers. Okay. So the ratio is 1:1:1:1. Now children, will go on to some modern application of genetics. So we'll talk about 2 modern application. One is genetic engineering. So what happens in genetic engineering is the genetic constitution of an organism, that is most of the times bacterium, is altered. So we alter the genetic constitution of an organism using genetic engineering. So how do we alter it? By introducing new genes into the chromosome. So we artificially introduced new genes into the chromosome. Here you can see that this is a bacterial cell. Here artificial genes are introduced into it, and then the genetically modified organism, that is GMO, that this is now genetically modified, thus produced, is then grown to multiply. It is allowed to multiply, and we know that bacteria can multiply very faster. So this is allowed to multiply, and the gene product is obtained in large quantities, and we can obtain the same gene product in a very large quantity, and then we can use this gene. Okay. Now the hormone insulin was produced this way. So insulin producing genes of a mammal, because mammals produce insulin like we. So insulin producing gene of a mammal like human beings were taken and introduced into bacteria, okay, which have been made to produce it, and then bacteria produce these genes in large quantities. So this is how we obtain insulin in large quantities artificially. Next is genetic counseling. Now children, what happens in genetic counseling is newly married couples are advised by their doctors or specialists regarding the possibilities of any undesirable trait which their children might inherit. So they or their forefathers might be having some trait which the children can inherit and which is dangerous for their lives, so and which is unwanted, undesired trait. So what do these specialists do? They sit with the newly married couples and decide on the possibilities of inheriting such traits. So what does the process include? They interpretation of family and medical histories to assess the chance of the disease occurrence or reoccurrence, education about inheritance, testing, management, prevention and resources, and then counseling to promote informed choices. Now they have the choice that whether they want to go for it or not, and adaptation to the risk or condition. So you should remember that the few genetic diseases are there for which genetic counseling is required are hemophilia. I'll teach you hemophilia in the next part of this video in detail, because this is a sex inherited disease, sex linked disease. So in hemophilia, the bleeding doesn't stop. Then thalassemia and sickle cell anemia, which deals with the deformities in the shape of the cells, okay, blood cells. So these are few of the genetic diseases for which genetic counseling is required. Today we are on part 2 of genetics. So in this video lesson we'll be talking about sex-linked inheritance and also mutation. So let's go and read in detail about the sex-linked inheritance. Now children, there are 2 diseases that we will be studying. So out of that, one is color blindness. So a color blind person cannot distinguish between red and green colors. So these colors appear grey to them. So a person who is suffering from color blindness is not totally blind, but he cannot distinguish between 2 colors, that is between red and green. So here if you can see this diagram, okay, so this is the normal vision. Okay, a person can see, a person who is not colorblind can see all these colors, but this is a vision of a colorblind person. Here you can see this green color and red color seems to be grey for him, right? So how does this color blindness occur? This color blindness is a sex-linked inheritance. Now we'll do an exercise to check whether you are colorblind or not. Can you read the numbers that are there inside? Okay. So it is number 25, 45, 6, 29, 56 and 8. And if you are not able to read these numbers, then you might be suffering from such disease. Now the second disease that we'll be talking about here is hemophilia. Now hemophilia is also a genetic disorder in which the sufferers are at the risk of bleeding to death. So even if there is a small injury or a small cut and the bleeding starts, the bleeding won't stop and they may bleed to death, okay, because in these person blood clotting doesn't happen. So this is also a genetically inherited disease. So this is also a sex-linked inherited disease. So both these diseases we'll be talking about now children, who are the sufferers in this disease? Homozygous recessive female and the recessive X-bearing male. In these two also, most of the time it is the males who are the sufferers, and very rare cases there are female. Okay. So what are homozygous recessive female and the recessive X-bearing male? Let us study. So here we are taking 3 cases of color blindness. Now see case 1. In case 1, there is a breeding between a normal mother. You know, in a female there are X and X. Okay, so normal mother is there and a color blind father is there. You know that in father X and Y chromosomes are there. Now on the X chromosome, okay, we have put a sign like this indicating that this is the carrier. Okay, so the color blind father is colorblind due to this X chromosome. Okay, so now from a normal mother we are taking X and X gametes, and from a blind father we are taking X naught and Y. Now when these gametes are fused, we get X naught. Here also we get X naught. Here we get XY, and here also we get XY. So here you can see that this is a normal male, this is a normal male. Okay, now these two are carrier females. Okay, they are carrying this. Okay, they are carrying this, but they are not affected because in this disease it is the male mostly that is affected. So none of the children is color blind, but the daughter are the carriers of defective allele. So these two, that is the daughters, are the carriers. Now let's go ahead. Now if this carrier mother, okay, in case 2 what we have done, we have taken this carrier mother who is not color blind. Okay, so we have taken X naught here, and again the normal father X and Y. Okay, so here we will get XX, here we will get X naught X, here we will get XY, and here we will get X naught Y. Now children, this is a normal female and this is a normal male. Okay, now this is a carrier female. Okay, so this is a carrier female, but here you can see this is a male who's having this X chromosome, okay, which is the affective allele. So this is the infected person. Okay, so this is the infected male. So here you can see that 50% sons will be colorblind. Okay, there are, there are 2 types. So these are the sons. So out of them 50% will be colorblind and 50% are normal. These are normal. Okay. Now 50% of the female are normal, whereas 50% are carrier. So here you can see that a colorblind father transmits the disorder to the grandson. So here was the colorblind father. Okay, he did not transmit the disease here, but he transmitted the disease here. Okay, so of this father, these were the kids, and out of this kid, okay, the carrier mother, this male was infected. So he is the grandfather and he is the grandson. Right? So grandfather, colorblind father has transmitted it to the grandson. Now here you can see that again this carrier mother, we have taken XX naught, and now a color blind father. We have taken a color blind father here. You can see X naught Y. Okay. So the gametes of carrier mother are written here and a color blind father is written here. X naught Y. Now X naught, these two gametes will combine to form X naught. This will combine to form XY, and these two will combine to form X naught Y. Now here you can see that this is a normal male. Okay, so normal male is 25%, right? Now this is a color blind, that is infected male. So 25% is color blind male. Okay, now this is the carrier female. Okay, because only one X is containing this. Now here both are containing this. Okay, so she is a color blind female. Now children, I told you in the starting that in this type of diseases mostly male are affected and there are very less chances of a female being affected. So here now after a long journey we can see a colorblind female also. Now children, one more thing to be noted here in case 2 is that a male child got this disease from a carrier mother who is otherwise not infected. So here a mother is giving the diseases, the mother is giving the disease to the son. Okay, now in this case a father who's a colorblind father, okay, the father is giving it to daughter through a carrier mother. So this type of inheritance is also known as crisscross inheritance, because here mother is giving to the son, the carrier mother is giving to the son. Here the colorblind father is making the daughter colorblind through a carrier mother. So female to male and male to female. That is why it is also known as crisscross inheritance. I hope you understood all the 3 cases very well, and I hope you understood that X naught is a carrier only because only one gene is affected, whereas X naught is a colorblind female. Now XY is a normal male, whereas X naught Y is a colorblind male. So I already told you about crisscross inheritance, that mother to son and father to daughter. So inheritance of X-linked genes as in color blindness and hemophilia. So whatever we did right now goes true for hemophilia also. The same diagram you have to draw. Now this is called crisscross inheritance. Now this I have already told you, because the son may get it from the otherwise normal but carrier mother, and a colorblind father may pass it to the daughter, making her colorblind if the mother is a carrier, as in case 3. Now there is also something known as Y-linked inheritance. Earlier what we did was X-linked inheritance, okay, because X naught was the carrier. Here Y-linked inheritance is also there, but this is not in your portion. I am giving it just for more information. The Y-linked inheritance is a trait which occurs only in males, as this Y chromosome is only in males. As Y chromosomes only males have example traits such as hairy pinna. So the pinna, the outer covering, contains hair. The hair grows there. This disease is known as hypertrichosis, and this is found in men only. So Y-linked inheritance is direct. This is not crisscross. This is direct. That is father to son. Okay. Earlier in X-linked inheritance we studied that mother was giving to son, and also the colorblind father was making his daughter colorblind due to the carrier mother. But here the Y-linked inheritance is direct, that is father to son. So there are no females involved. Now let us read what is mutation. Now mutation is a sudden change in one or more genes or in number or structure of chromosome. So either there is a defect in gene or the defect in chromosome or the change in number of chromosomes. So mutations are of 2 types, gene mutation and chromosomal mutation. So here in gene mutation you can see there are different types of damages. Here you can see that the double strand breaks. Here if you want to see the chemical bond between the neighboring nucleotides. So if this is the nucleotide and this is the nucleotide, you can see the formation of a bond. Now here there is chemical modification of the nucleotide. It's a chemical modification here, A, T, C, G. Okay. Now there is a chemical linkage of two strands. You can see this one strand and another strand here. So the linkage, the chemical linkage occurs. Because of all of this, we can see the mutation in genes. Now let us look at the chromosomal mutation. Here you can see that there is a deletion. So B gets deleted. So there is deletion, there is duplication. You can see there is two B's here. Okay, there you can see there is an inversion and there is a translocation. So these are the different types of chromosomal mutation. It is not there in your course in detail. Just to give you an idea what chromosomal mutation is all about. Now, under the mutation, we have a disease known as sickle cell anemia. So this is a blood disease which is caused by gene mutation. So here there is a change in DNA, and because of that, the red blood cell becomes sickle shaped. So here you can see what happens in sickle cell anemia is, this is the normal red blood cells flowing through the capillaries, okay, and if you see the normal hemoglobin, the cross section is like this, but these are the sickle cells, RBCs, sickle cell shaped RBCs, okay. So what do? They tend to block the blood flow, and also when you see inside it, okay, they form strands, that you can see the formation of strands here. Okay. So what is sickle cell anemia? Sickle cell anemia is a genetic disease, is a blood disease caused due to gene mutation, and in this the shape of the red blood cell changes like a sickle. Now you all must have heard about the atomic explosion during World War II in Japan. The cities like Hiroshima and Nagasaki, there were atomic bombardings done. Okay. Now this led to deformities in the bodies of plants and animals which are still persisting there. Okay. You all must have read in your history class that what happened during the Hiroshima and Nagasaki time. So you should know that gene structure mutation happens or occurs by radioactive radiations. Okay. So these are also mutations you should know about. On part 3 of genetics. Now in this video lesson we'll be talking about Mendel's experiment on inheritance. We'll talk about what a monohybrid cross and how to derive it, and what a dihybrid cross and how to draw the Punnett diagrams. So let's get started with these very important topics of genetics, and I'll make sure that you are able to answer all the questions related to these topics. So children, you must know first who was Mendel. So Gregor John Mendel was the father of genetics. Okay, his early education was in a monastery, and then he went to Vienna for his further studies of science and maths. Now he always wanted to become a teacher. Unfortunately, he failed in the examination of becoming a teacher. But what he discovered later, what he found later, made him the teacher of teachers like us. Okay. So when he returned back to his monastery, he conducted a lot of experiments, the breeding experiments on garden pea, which was there in the beautiful garden of the monastery. Okay. So out of his sheer interest, he started conducting breeding experiments on pea plants. Okay. And his findings became a milestone in biology. So let's go ahead and find out. Now, he conducted the experiments on garden pea, as I told you, whose scientific name is Pisum sativum. Students, you should always remember that when you are writing the scientific name, okay, the second name, okay, it should be in small. So don't write S capital. So he conducted study on Pisum sativum. Now, why he chose Pisum sativum only? Okay, he selected it for a particular reason. One, the pea plants have a large number of contrasting characters. Means you can find pea plants purple color and white color. The seeds also have variations. The height also have variations. So we'll study about these variations. So you should remember that it has a large number of contrasting characters, and the different physical characters were easy to recognize and study. That is why he chose pea plant. Next, the varieties were available in pure forms that bred true. What do you mean by pure forms that bred true? It means they produce the same type generation after generation, exactly same type. The varieties that were found were pure. They were not hybrid of two different varieties. Now they are self-pollinating, means in one plant self-pollination can occur. Also cross-pollination can easily be performed by removing certain parts of the plants. So it was self-pollinating and also cross-pollination is very much possible. Then the flowers of this plant are bisexual. It has female as well as male parts, so bisexual. They have a shorter lifespan, which makes it very suitable for carrying out the experiments, and the plants are easier to maintain because it lives for a shorter period of time. It is easy to maintain and easy to conduct experiments. Also, they have a very good resistance to disease and they have an ideal survival rate. So these are the characteristics of a pea plant which makes it ideal for Mendel's experiment. Now he took seven characteristics of pea plant. Let's look at the first characteristic was about seed, that the shape of the seed is round as well as wrinkled. So these two variations are found in different plants. Then the second is the color of the cotyledons of the seeds. Okay, it can be yellow or it can be green. Now let's look at the third characteristic. The color of the flower can be white or violet. So these two variations are there. Then the fourth characteristic that he studied was the shape of the pod. It can be full, it can be straight pod, and it can be constricted pod. Okay, smooth or constricted you can say. Then the color of pod was the fifth characteristic, which is the pod can be green or the pod can be yellow. The sixth characteristic that he studied was the placement of the flowers. Okay, here you can see it is the flowers are placed axially, whereas here it is on the terminal at the end. Okay, here they are at the end and here they are in between. The next is size. Here you can see the plant is tall and here it is short or dwarf. I'll enlarge this and show you exactly the terminal and axial pods. Here you can see the flowers are in between and here they are at terminals. Okay. So these were the seven characteristics that Mendel chose to work upon. Now let us look at what monohybrid cross is. So here I am taking one of the characteristics, that is the color of the flower. You can take any of the characteristic and work out the monohybrid and dihybrid cross. So here I have taken the color. So you should know that in color this one is the dominant character. The purple color is the dominant character. Okay. So capital B suggests that it is a true breed. Okay. Both are, it is homozygous dominant. And small b is for recessive because white color is recessive. Now children, before going further, I should also tell you that you should read the characteristics or all the seven characteristics very carefully. You should know which is dominant and which is recessive in it. So in this case of flower color, I am telling you that this is dominant and this is recessive. So you should know which are dominant and which are recessive to work out these diagrams. Okay. So now this is homozygous. Why homozygous? Because both are same and it is also dominant. So this is homozygous. I'm so sorry. Dominant. Okay. And this is also homozygous. But this is recessive. So now this is producing capital B gametes and this is producing small b. Okay. So I'll show you the combination in Punnett square. So if I take this capital B here of this purple flower, and for this white flower if I take small b here and try to work out the combination. So here it will be capital B small b. Here it will be capital B small b, same capital B small b, and now these two also gives you capital B small b. So if you see the genotype, all are same. Also if you see the phenotype, in all of them, okay, purple color is dominant, one dominant character is there. So all of them becomes the same genotype that is BB. Genotype is what type of genes are there. I already told you in my last video. And the phenotype, that is the physical appearance, the gene that is expressed, is also all of them are purple in color. So if you find out the genotype and phenotype ratio, the genotype ratio is 1:1:1:1, and also the phenotype ratio is the same. Now that was the F1 generation, that is the first generation. Now we are coming on to the second generation. So here you can see that in the first generation, all the plants that were formed were capital B, small b, right? So if I take two such plants, capital B, small b and capital B, small b. So here you can see this is the first plant which are having the gametes capital B, small b, and this is the second capital B, small b. Okay, so we have taken the pollen of this and the pistil of this. Okay, so male and female we have taken. Now you can see these two combination will give a capital B, capital B. This is what? Homozygous dominant. Here when you see that we have taken this small b and this capital B, this is heterozygous. Why? Because both are different. So heterozygous, and it is also dominant. So I am writing D. Okay. So this was homozygous dominant. This is heterozygous dominant. So both of them are showing dominance. So both of them are purple color. Here also you can see one capital B and one small b. So this is also heterozygous, and here also you can see one dominant. So heterozygous dominant. But here you can see one small b and one small b. So both are small b. So it becomes homozygous. But here you should see that both are recessive. So this is homozygous recessive. Okay. So if you see now the phenotype, that is the physical appearance what we are seeing, you can see that there are 3 purple and 1 white. So the phenotype ratio is 3:1. Whereas the genotype, now here you can see one is capital B, but there are 2 heterozygous dominant, capital B and small b. Okay, so one is capital B, then there are 2 capital B, small b, and then there is a homozygous recessive, that is both are small b. So this ratio is 1:2:1. Now children, this is very important to remember this phenotype and genotype ratio of monohybrid cross. Okay. Now the ratio remains same, that is for phenotype it is 3:1 and genotype it is 1:2:1 for any of the characteristics that you take. So here we have taken the color of the flower. You can take any of the characteristic and work out, you will get the same ratio. So that is your homework for today. You will work out for different combinations. Now we are going on to dihybrid cross. Now in dihybrid cross, what did Mendel do? He took 2 characteristics simultaneously. Here you can see that Mendel has taken the color of the seed as yellow and green, and he has taken the shape of the seed as round and wrinkled. So this seed, if you see, is yellow. Now in the color of the seed, okay, there are 2 variations, that is yellow and green. Okay, out of which you should remember that yellow is dominant. That is why I have taken yellow dominant, so capital Y, and green is recessive, so I have taken small y because I have to take the same letters. Okay, so capital Y represents yellow color which is dominant, small y represents green color which is recessive. Now capital R represents the shape of the seed, that is round. Now in the shape of the seed there are 2 variations, round and wrinkled. So round is dominant, you should remember, that is capital R means round and small r means wrinkled. Right? So it's taken 2 variations, okay, 2 characteristics. So when you compute the F1 generation, the first generation, you will see that when you breed these 2, you will get all the 4 generations which are having this constitution. Right? Now if we take 2 such parents, capital Y, capital R, small r. Two such parents, we will work out the F2 generation. Now here, what they have taken, see here capital Y, capital R, small r. Okay, there also. Now, if you see capital Y and capital R, then small y and capital R. So, small y, capital R, then they have taken the combination capital Y, small r. So, capital Y, small r, and then both small y, small r. Right? The same combinations you will take here for the other parent, and then you crossbreed. So, when you see capital Y, capital R and capital Y, capital R here, it forms YYRR, in which all are capital. So what does this seed mean? This capital Y and capital R mean? It means that the seed is yellow and round. Okay. So when you work out everything here, I will show you. This is capital Y. Okay. So in the color, the capital Y denotes that it is yellow, and you have got one R which denotes it is round. Okay. This is also yellow and round, and this is also yellow and round. But here if you see that these in the color, both are recessive. So it will show green color, and R shape mein R is dominant, so you will see round. Okay. Now in this, if you see, color is yellow only, okay, because Y is dominant, but here you can see both of them are small, so the shape is wrinkled. Now here if you see, in the color also recessive is there, and in shape also this is recessive. So this is green and wrinkled. Okay. So once you work out this, then you find out the genotype and the phenotype. Okay. Now let us see the phenotype first. Okay. In the phenotype is what you see. Okay. So if you see yellow and round, how many yellow and round are there? 1, 2, 3, 4, 5, 6, 7, 8 and 9. So there are 9 yellow and round. So you write 9 here. Okay. Then when you see green and round, so this is green and round, 1, 2, 3. So there are 3 green and round. Then yellow and wrinkled. So where are yellow and wrinkled? This is 1 yellow and wrinkled. This is 2 yellow and wrinkled, and there is 1 more yellow and wrinkled. So this is also again 3. And you will see that both the recessive traits, that is wrinkled and green, is seen only in 1. So 1. So that is why the phenotype ratio becomes 9:3:3:1. Remember that whenever you are writing the ratio, above it you should also write which character it is, so that you are very sure that you have not mixed the variations. It is easier for the examiner also to check. Now children, we have worked out the phenotype, but genotype of this is very complex and it will not be asked to you in the examination for your portion. So I hope you understood the dihybrid cross also very well. Two things you have to remember in this. It is very important to write genotype and phenotype ratio. Also in the 7 characters, you should remember which one is dominant and which one is recessive. Otherwise, you will not be able to form the generations F1 and F2 generations, and you will not be able to draw the Punnett diagrams. So please remember the 7 characteristics. Please remember which one is dominant and which one is recessive. Try to work out different monohybrid and dihybrid crosses on your own. If you have any doubts, please write in the comment section. I'll be ready to help. And we are on part 4 of genetics, which is a very important topic in biology for all the boards, for all the students. And in this video lesson, we'll be talking about Mendel's law of inheritance and its applications. Last lecture, we studied all the experiments that Mendel conducted. We studied about the pea plant. We studied about dihybrid and monohybrid cross. Now in this, we will study the laws that he formulated using them. Now Mendel formulated few laws which are known as laws of inheritance. The first law is law of dominance. Second is law of segregation. And third is law of independent assortment. So moving on to our first law, that is law of dominance. First we'll read the law. Out of a pair of contrasting characters present together, only one is able to express itself while the other remains suppressed. The one that expresses is the dominant character and the one unexpressed is the recessive. The recessive character can express only when the pair consists of both recessive, that is homozygous recessive. Now children, to understand this very well, let us consider an example of a classroom. Now in a classroom there are a variety of students, okay. Some are very outspoken, so whenever a teacher asks questions or whenever they have a doubt, they'll just raise their hand and stand up and they are very dominant in the class. Whereas there are few who are very shy, so we'll take them as recessive. So out of a pair of contrasting characters present together, only one is able to express itself while the other remains suppressed. So only one or two child will get up and answer or will ask questions, whereas the other will remain suppressed. One that expresses is the dominant character, the student who is asking question is a dominant one, and the other that are unexpressed, they don't express their feelings, are recessive. Now students, suppose these 2 or 3 students who are dominant goes out of the class, okay, they are not in the class, then only the students who are recessive, who are unexpressed, okay, they can get the confidence to raise their hand and ask question, okay, or answer any question. So when are they able to express? When there are no dominant people, okay. So they can express only when the pair consists of both the recessive. So only those students are there who are recessive already. In their presence only they can express themselves. So I hope now due to this analogy you are able to understand the law of dominance. So here if you see, we have studied this in our last video, okay, that if we take 2 heterozygous cross, that is capital B small b and here also capital B small b. You remember this was the first generation. So when we take these two, okay, capital B small b and breed them. So here you can see this capital B, here both are dominant. So that is why this is able to express its color, that is purple color. In the color, if you remember, purple is the dominant and white is the recessive. So we take capital B for purple because it is purple or violet, you can say, because it is dominant, and small b for white because it is recessive. We can take only one letter, that is why we have taken capital B and small b. So capital for the one who is dominant and small for the one who is recessive. So here if you see, both are dominant, okay. Here you can see one is dominant and one is recessive. So who will answer? The capital one, the dominant one. So here capital B, so again purple. Here also you can see capital B, small b, but here if you see, both are recessive. So now the dominant one is not there. So both are recessive. So that is why he is able to express his color, that is white. So when both of them are recessive, it will express its color. But in the presence of a dominant and a recessive gene, who will express? The dominant one. So this is the law of dominance. Now let's go to the second law, which is law of segregation. Now this law is also called as law of purity of gametes. So first let us read. The 2 members of the pair of factors, okay, separate during the formation of gametes. So if there is a pair of factors, they will separate during the formation of gamete. They do not blend but segregate or separate into different gametes, and the gametes combine together then by random fusion at the time of zygote formation. So if you see here, if you want to understand the law of segregation, just look here. Here you can see 3 pairs of chromosome with the factors that we require. So now what happens? They segregate, okay, and then combine randomly. You can see the gametes in which they have first segregated and then combined randomly, okay. So the 2 copies of each gene, separate and end up in different gametes. Now here if you try to understand the same thing, this was capital B, small b, right? So this separates and form capital B and small b differently. This also separates and forms different gametes, and then they recombine. So here you can see they are recombined like this. So this is law of segregation. Okay. Now the law of segregation is also known as law of purity of gametes. So this is the other name of this law, law of purity of gametes. Why this law is also known as law of purity of gametes? Because a single gamete carries only a recessive or a dominant allele. So a single gamete carries only either a dominant or a recessive allele, but not both the alleles. So single one contains only one dominant or one recessive allele. It does not contain both the alleles. That is why it is also known as law of purity of gametes. I hope you understood law of segregation, which says that two members of a pair of factors first separate together during the formation of gametes, and then they do not blend, but they segregate or separate. So capital B and small b separate, and then they combine together by random fusion, okay, at the time of zygote formation. Now let's go on to our third and last law, which is law of independent assortment. Okay, they are assorted independently. So we saw in the dihybrid cross of Mendel's experiment that he had taken two characteristics, right? So here, when there are two pairs of contrasting characters, the distribution of the members of one pair into the gametes is independent of the distribution of the other pair. So what Mendel had taken, for example, two different varieties, yellow and round and green and wrinkled. Yellow, the color of the seed is a different character, and the shape of the seed, that is whether it is round or constricted or wrinkled, is another character. So there were two contrasting characters. So when there are two pairs of contrasting characters, the distribution of members of one pair into the gamete is independent of the distribution of other pair. So when you are exchanging the yellow, okay, the color, then the shape does not interfere, okay. So the distribution of the members of one pair, so what is the one pair? The color, okay, into the gametes is independent of the distribution of the other pair. The other pair can be shape, okay. So Mendel experiment showed that the alleles for round or wrinkled piece were inherited independently from the alleles for yellow or green piece. The alleles for both the characters were inherited independently, and that is why this law is known as law of independent assortment. So I hope you understood all the three laws. Now let's go ahead with the applications of Mendel's law. Now the study of these laws, the understanding of these laws helps us to predict about the new combinations in the progeny of hybrids, and this can be useful to solve various genetic problems, whether it be genetic problems in plants or in human beings or animals. Then it is important to plant and animal breeders for producing a better breeds, because if he knows and understands all the laws very well, he will be able to breed perfectly. And hence new types of plants with new combinations of useful characters can be produced by hybridization. That is why the study of Mendelian laws, that is laws of inheritance, is very, very important and crucial. So children, this was all about the laws of inheritance and few applications of laws of inheritance. I hope you liked the video. If you have liked the video, give it a thumbs up and write in the comment box, and also share our videos with your friends. If you have yet not subscribed to us, please subscribe right now, and don't forget to tap on the bell icon to receive all the important and interesting videos. So keep the learning on. Thank you.